When you get sick, you need your immune system to help you feel better. That's what happens when you get a cut or when you get a virus. But what about in the brain? In diseases of memory loss, like Alzheimer's disease, which is the most common form of progressive dementia, people knew that the immune system would respond to this disease, but they didn't really understand that this response can actually modulate whether or not someone gets Alzheimer's disease, until recent genetic studies were able to show that the mutations associated with increasing risk of this disease affect the immune system. The problem now is that we don't really know what they're doing. Do people with Alzheimer's disease have a hyperreactive immune system, or is their immune system too suppressed? So in order to figure this out, I'm studying two of these new Alzheimer's disease mutations. One associated with increased risk for Alzheimer's disease, and a protective mutation associated with decreased risk for Alzheimer's disease. What I've found is that the mutations that are increasing risk for Alzheimer's disease make it so that the immune system is not responding to the disease. And likewise, the protective mutations, or someone's less likely to get Alzheimer's disease, are actually making the cells better at responding and clearing the disease. So this is really exciting data because it shows what we need to do in creating a vision for new drugs and new therapeutics against Alzheimer's disease. We need to be able to reactivate the immune system so that things are acting more like these protective mutations and giving the immune system a chance to fight back against Alzheimer's disease. Thank you.
Hello everyone, I hope you're all staying sane and healthy. I'm sorry that we couldn't be together for our normal ARCS dinner where I could have presented this work to you in person, but I'm excited to still get the opportunity to share with you the work that I've been doing during my Ph.D. program in the lab of Ziva Wunderlich, working to understand how different genetic systems are able to achieve normal embryonic development. And so with that, I'm going to go ahead and share with you my presentation so you can see that. So when we think of genetics and DNA, we often think of genes that encode a protein that performs some function. But the large majority of our genome, 99% of our DNA, actually does not encode proteins. And so when we first realized this, there was kind of this question in the field, why would so much of our DNA consist of this junk DNA? But biology is quite clever, and over the past couple of decades, it's become clear that this non-coding DNA is anything but junk. It often plays very critical roles. Much of this non-coding DNA are regions that actually control the activity of protein-coding genes. One critical class of this regulatory DNA are enhancers. And so enhancers are sequences of DNA that control the activity of target genes. And enhancers work by binding key effector molecules called transcription factors that I'm showing here as the different colored circles. And when the right transcription factors are available, an enhancer will turn on its target gene at a particular time or place. And so that's what we're looking at here. We're looking at a developing mouse embryo where gene expression is being controlled by an enhancer, and that's what we're looking at in blue, where gene expression is being told to be turned on in the spinal cord by a particular enhancer. And so by controlling gene expression in this manner, enhancers play critical roles in development. It is through regulation by enhancers that, for example, a muscle cell knows to become a muscle cell, while a nerve cell knows to become a nerve cell. We can see just how important enhancers and other non-coding regions of DNA are simply by the fact that 93% of the disease-related mutations that we know about occur in these non-coding regions of the genome. And so my project focuses on understanding a particular kind of enhancer that seems to be quite pervasive in developmental systems. These are called shadow enhancers. Shadow enhancers are groups of multiple enhancers that all control the same target gene and turn on that target gene in a very similar pattern. And these shadow enhancers have been shown to be critical for proper development under conditions of stress, but no one knows exactly how or why how these shadow enhancers are able to function. But a key characteristic of shadow enhancers that I hypothesize is critical for their function and development is that the individual member enhancers of these shadow enhancers groups bind different transcription factor, those effector molecules, that allow them to activate their target gene. So that's where we're looking at this example here, where our one enhancer called proximal is activated by these transcription factor molecules, while this second enhancer, the distal enhancer, is activated by these two different transcription factor molecules. Yet both of these enhancers are able to drive a similar pattern of expression of their target gene. And we think it is through this, what we're calling a separation of inputs, that shadow enhancers are able to buffer against fluctuations in the levels of these transcription factor molecules, and therefore drive more stable levels of expression of their target gene. And so in order to actually test this hypothesis and assess how shadow enhancers are functioning to ensure robust development, I visualized the activity of these enhancers in living Drosophila embryos using fluorescent microscopy. And that's kind of the pipeline we're looking at here. And this is an example of some of my imaging runs. And by tracking the activity of these different enhancers across developmental time, I can learn about how they each function to control gene expression. And so with help from my ARCS funding, I've made significant progress on this project. I actually have a paper that we're currently in the process of resubmitting that shows indeed, kind of circled here, a shadow enhancer pair drives more stable gene expression than single enhancers or duplicated enhancers. And this is true even across a wide range of temperatures. So this is really exciting finding that was a really nice conclusion to this project. And so my findings may underlie why exactly shadow enhancers are so common in developmental systems. And these findings might help us better understand the mechanisms by which development not only functions normally, but also why it can sometimes go awry, and hopefully inform future efforts to prevent some of these developmental issues. And so with that, I'd like to thank everyone in the Wonderlic Lab for all of their help and support, as well as a big thank you to our collaborators in this project, as well as obviously a huge thank you to ARCS for all of their help and support during my time in my PhD program. The funding from ARCS, the ARCS Fellowship actually has enabled me to attend multiple conferences where I've been able to present this work. So I really can't thank ARCS enough for their support. And I thank you all for listening. And again, I hope you're all staying sane and safe, and I hope to see you all again in person very soon.